Hi guys, so today we are talking about cell division, and we're finally talking about this last objective right here, that we are evaluating the methods of cell division and exploring and understanding the process of growth and development and why it is so important for organisms. This is our last chapter in the current unit that we are working on, and finally we will be getting into genetics. So just some easy vocab. I do have a different worksheet that we are going to use to take notes in class. So when you come back, make sure you get that from me. I've also posted the answer key to this note sheet in Google Classroom for you. So the cell cycle is a pattern of growth, DNA replication, and cell division that occurs within the eukaryotic cell. Mitosis is how our body cells are going to divide its nucleus and all of its contents. And then cytokinesis is how the cytoplasm for the cell is going to divide. The cell cycle has very distinct growth and reproductive phases, as well as making sure it is accomplishing its everyday job. And there are four main stages to the cell cycle. Gap 1, right here, which is also called G1, synthesis, which is also called S, Gap 2, which is called G2, and then the mitosis phase, which is M, and that involves both mitosis and cytokinesis. The names are given from early studies of cell division and how the cell is actually operating in that point in time. What happened was they found that during what we call Gap 1, there was no real functions happening. Synthesis, the DNA was replicated. Gap 2, there was nothing really happening. And then cell division, mitosis. Gap 1 is when the cell is carrying out its everyday job. It's growing, it's doing it, whatever its function is. And at the end of Gap 1, the cell must pass a critical checkpoint in order to make sure that it is ready to proceed into the synthesis phase. This checkpoint makes sure that the cell has the proper nutrients it needs to move forward, it's adequate in size, and its DNA is relatively undamaged. In synthesis, or DNA replication, the cell is physically making a copy of its DNA. It's not doing anything else during this point. It's the nucleus's job to take over. Human cells have 46 chromosomes in them. And when you multiply that by 2 during synthesis, we end up with 92 chromosomes in our cells. That's crazy. But this makes sure that every cell has the proper number of chromosomes in it moving forward. By the end of synthesis, the cell has 2 complete sets of DNA. There is a minor checkpoint here, but it's performed during the DNA replication phase. And as we'll talk about that, there is an enzyme that works in DNA replication that actually makes sure that it is proofreading itself and that there are no errors. In Gap 2, it's the third stage of the cell cycle. This is when the cell is continuing to carry out its normal activities. It has to continue to grow, and it must pass a critical checkpoint at the end of Gap 2 before division can happen. This checkpoint makes sure that the cell is double its original size and has undamaged DNA. We can then enter mitosis, and this happens in 2 phases. The first being the cell division, and the second, the cytoplasm division. The division of the cell and all of its contents and its cytoplasm is what completes mitosis. The result is 2 genetically identical daughter cells that are identical to the original cell. This is because we want skin cells to constantly replicate skin cells. We do not all of a sudden want a liver cell to be growing on our shoulder. That means something isn't working properly. So we want our cells to be genetically identical in the end. Cells do divide at different rates though, and the rate of cell division depends on the type of cell that it is. This chart shows the average human cell type. So average adult human cell type, and it changes with age. Skin cells in babies and up and through probably, it's usually like 16, 17, skin cells can turn over anywhere from 24 to 72 hours. But as we get into adult age, that skin turnover is 2 weeks, which is why we age. Red blood cells don't turn over quite as often. They turn over about every 4 months. Your liver cell turns over every 300 to 500 days. The internal lining of your intestines turns over every 4 to 5 days, but the external lining, the muscle, turns over every 16 years. The younger you are, the embryo, the children, the cell division is much more rapid. They're still growing and developing, but once we stop growing, these times actually stretch out much more. Some cells remain in a stage called G0 or G0. They rarely ever enter cell division and can stay in G0 for 16 years. Now, just because our skin turns over every 2 weeks doesn't mean every single cell in your body that is a skin cell turns over every 2 weeks. That would be really bad. But if you get a cut, that timeline is going to be sped up so we heal faster. But for 16 years, these cells remain in G0 before they are ready to go into G1 and then go through the cell cycle and then re-enter G0 because they're not needed in the cell cycle at that point in time. Cell size is very limited. Cells have both upper and lower limits for their size, which means they can't be too big, but they can't be too small. Every cell must contain all necessary materials and organelles in order to suffice life as a cell. And that doesn't mean that every single cell has every single organelle. Red blood cells don't contain a nucleus once they mature because they need more room to do their job. So the white blood cell takes the nucleus. All cells do have to maintain an appropriate size, growth, and division. So all three of those things must be coordinated. Cells have to double in size before they divide to make sure that the cell that is created is appropriately sized for the function of that cell. We're going to stop here. We will go into mitosis and cytokinesis next time.